does not have antigen receptor. DNA, RNA, they don't have antigen receptor. Only protein and glycoprotein, they can be antigens. So, usually proteins are polysaccharide, lipid and nucleic acid only when combined with the carrier protein or polysaccharide. Only when in the form of combined or in the form of coupled form, they can have antigen receptor. Non-microbial, non-cell, and these are the properties of antigen. See, three properties. Has a configuration or determinant. Okay, so as you know, epitope is there. What is epitope? It is antigenic determinant. Okay, antigenic determinant, that means only that portion of the antigen is responsible for its antigen. Okay, then the capacity to produce corresponding antibodies, as I have already told you, then the ability to react specifically with the antibody molecule. So these three properties are there in the antibody. Now there are other terms are also there. Immunogen, which reduce cellular immunity. Antigenicity, ability of a substance to lock with the antibody molecule. Immunogenicity, the property of immunogen to generate immune response. Hapsin, it is a half antigen. It can bind with the antibody but cannot generate the immune response. Then allergen. What are allergens? They cause detrimental reaction or allergic reaction in the one's body. Then tolerogen, capable of making lymphocyte or immunocompetent cells unresponsive to the inducing substance. If its molecular form is changed, it can become an immunogen. Otherwise, it is not immunogen. It can be tolerated. It can be tolerated by the body. That's why it is tolerated. But if you change its configuration, so what happens? It can become an immunogen. Now the next is classification of antigens. There are different methods or different types of classification. First is on the basis of source. They can be exogenous, they can be endogenous on the basis of their source. Then second is synthetic or natural. Obviously, natural antigen is generally they are very complex type and always made up of more than one type, 15 to 20 building blocks of protein and 3 to 5 in carbohydrates. Then third is conventional and purified. Conventional means impure, okay, a combination, okay, and second is purified, means if you purified toxin, so that is purified antigen, and if you just give the microorganism, that is conventional antigen. Okay. Then recombinant antigen and anti-isotype antigen. Recombinant, it is a product of recombinant technology, and anti-isotypic antigen, that is the antigen which is specific or which is specifically bind with the ideal type of antibody. Okay, as you know, there are different types of different classes, not classes, different types of antibodies. They are determinants: allotype, isotypes, and isotype. So, these are the antigens which are specific to ideal type of antibody. Next is cell surface antigen derived from a variety of cells. They are present on the RBCs and WBCs. They are membrane bound. Okay, that's why they are known as cell surface antigen, because they are present on the cell wall or cell membrane of the cell. Then autoantigens, that means they are self antigens, and when the body reacts, means if the recognition capacity of the immune system is lost, so that leads to autoimmunity. And when this body responds to its own antigen, so those antigens are known as autoantigens, just as in case of myasthenia gravis, just in the case of hydroxycosis, just in the case of glomerulonephritis. Okay, so those receptors, they act as autoantigens. Then tumor antigen and super response generally, and they induce B and T cell both, and actually these antigens, they bind simultaneously with the T cell receptor and class 2 MHC simultaneously, but outside the peptide binding. Okay, yes. These antigens, they bind simultaneously with TCR, T cell receptor, and class 2 MHC, but just outside the peptide binding tract. That means they are not internalized and degraded by antigen presenting cells, and they can produce 15 to 25 times more rapid response than the normal antigen. And that, because of this property, they create very harmful allergic reaction inside the body, like eczema. Okay, like eczema, like eczema and other pathological conditions. So that's why these are known as super antigens. Okay, because they produce 15 to 25% more rapid response than the normal antigen. Okay, and then they bind just like this. If this is T cell, okay, this is TCR, and this is class 2 MHC, okay, and this is TCR, so they bind here. As you know, this is peptide binding tract. Normal antigen should bind here. It generally binds here, but it binds just outside the peptide binding tract. Okay, and that's why it is known as super antigen. Now, the other one is, there are different molecules which are responsible for antigen recognition, but next is antigenic determinants, which is known as epitope. So there are epitope. It is a portion of antigen capable of binding firmly to the combining site of antibody on membrane receptor. Antigenetically specific and small. This small unit is called as epitope. It is a contour of three-dimensional structure, and epitope of B cell is different from A cell. Okay, it is not. These two are very different. Other than this, there are different types of antigens: haptenic and thymus dependent, thymus independent, and suppressor. But two types are very important, that is linear and conformational. Okay, linear means they are buried in the protein core, and when this protein is natured, only then they appear on the surface. And other one is conformational. That means they depend on the looping of the protein. Okay, and if this protein is denatured, what happens? They lose their antigenicity because they become separate. Okay, so they lose their antigenicity. First is both are they depend on the protein denaturation. First is linear, which appears only when the protein is denatured because it is present inside the protein core, and other one is disappear when the protein is denatured. Okay, and these are other haptenic, then thymus dependent and independent. Thymus dependent to T determinant, it requires MHC for stimulation of B cell with the help of T cell, and thymus independent is B determinant directly stimulates B cell. Then helper and suppressor determinants. Helper and suppressor determinants are distinct, which determine the balance between helper and suppressor T cell reactivity. is there is no antibody formation. But if this cell activates TH cell, this TH cell secretes cytokines, it stimulates B cell, and result is this antibody formation. Okay. Then fourth one is very important, that is cross reactivity determinant, and it is very important when you produce during monoclonal interaction. Okay? So identical determinant leading to cross reactions. So these type of antigens are responsible for cross reactivity. Do you know what is cross reactivity? One type of antigen can bind different type of antibody, or one kind of antibody can bind different types of antibody. So if this antigen is a multivalent antigen, that means it has 3 epitopes. First epitope A, second epitope B, third epitope C. Okay, that's why it is known as multivalent or trivalent. Now, what happens if you administer this antigen to the animal, experimental mice? So what happens? There is a production of 3 types of antibodies, mixture of antibodies. One antibody is specific to A, second is specific to B, and third is specific to C. So what happens? This type of, if you give this react this antibodies with antigen, so what happens? They can they can show cross reaction, and they can react with all these type of antigens. So this type of antigen is known as cross reactive determinant epitope or cross reactive determinant. Okay, now this is about antigen and epitope. Now, what are the specificity or what are the basis of specificity of antigen? This antigen must have some specificity. It must have some basis for specificity, like affinity, like avidity. What is affinity? It means these are factors which are responsible for antigen-antibody interaction. Okay. What is affinity? Oh, what is avidity? Basic specificity. So capacity to combine with one and discriminate from another. That is the first basis of specificity. Okay. Interaction of antigen with antibody is highly specific. That means that antigen is generally in normal condition, it interacts with the corresponding antibody. That's why it is highly specific. Then the overall configuration of antigen determinants is more important than the chemical structure for closeness of fit. Configuration is more important than chemical structure. Why? Because it requires closeness of fit. One epitope binds with paratope, and this binding should be very firm. Okay, so chemical structure is not very important, but overall configuration of the molecule is very important for antigen-antibody interaction. Okay. Besides these factors are also responsible overall configuration. Antigen can bind an area which may be larger or smaller than variable size of antibody, that means paratope of antibody. Then intermolecular forces, these are responsible for antigen-antibody interactions, that means van der Waals forces and hydrogen bonding, electrostatic attractions. These are responsible for binding. Then third is affinity and avidity. What is affinity? Binding capacity of antibody molecule with a univalent antigen. What do you mean by univalent antigen? Only one epitope. Okay, that bind with the multivalent antibody. That means binding at one side can enhance or generally enhance the binding of others. That is known as AFFINITY. These are diagrammatic representation of high affinity, moderate affinity and low affinity of antigen antibody interaction. If paratope and epitope are like this, so they can uniformly and show high affinity. This type of binding shows moderate affinity. If they are not corresponding or complementary to each other, so that binding leads to low affinity. These are valencies. Monovalent, only one. Bivalent. So one more. If monovalent antigen is there, so two antigens can bind with one antibody because antibody has two antigens in binding. And if it is divalent, so binding can be like this. If it is multivalent, that leads to precipitation and activation reaction, which can be visible, which can you can see by your eyes only. And if this reaction is in the form of immunodiffusion, that is also sometimes it is visible, but sometimes it is not visible because of the size of antigen antibody. Now the essential factors of antigenicity. These are essential factors. First and foremost is foreignness. That means it should be non-self. And self and non-self concept is introduced by Werner, as you know. Then molecular weight. A molecular weight of about 10,000, they are considered as the good antigen. If smaller, but sometimes smaller, like glucagon is also considered as a good antigen. Otherwise, very heavy and very low molecular weight antigens, they are not considered as good antigens. And you can enhance the antigenicity of low molecular weight molecule by adding adjuvants. From complete and incomplete adjuvants, you can enhance the antigenicity. Then molecular complexity, structural configuration, as I have already told you, that chemical structure is not important, but structural configuration is important. That means whether this antigen is multivalent or bivalent or monovalent, whether it has linear determinant or it has conformational determinant. Then composition of protein, which type of protein it is. Then disposition of grouping, position of groups. Okay. Then patterns of determinants, means whether they are repeating or linear, as I have told you, linear or repetitive pattern or conformational pattern, which type of repeating or alternating. Then adjuvant activity. If antigen is of low molecular weight and you want to enhance its affinity, you can add adjuvants, different types of adjuvants. Now dosage schedule is very, very important. Suppose you are designing and you are planning an experiment with a nice in vivo experiment, you want to inject the antigen. So dosage schedule is very important. Suppose you want to take immunosuppressed animal, so first you just inject the cortisone, and after injecting cortisone, after 72 hours of injecting cortisone, you can schedule your dosage. So that dosage should be, its first dose should be very low, it should have very low concentration of antigen. Otherwise what happens? That could be lethal. Okay. So first is very low with low volume, then again you can enhance antigen, and in this you can means to take booster dose. Means the last dose should after that you can sacrifice it as well. So dosage schedule should be very, very important. If you can, if antigen is not very toxic or not very harmful, you can give daily dosage. If it is actually, it is also is related with the route of injection. If you give IP, so that means what happens? If you give IP, so it spreads very fast. Okay. So it also related with the route, whether you are giving IP or IM, muscular or intravenous, which type of protein. So dosage schedule is very important. Then route and complex. Route and form of administration. Means whether you are giving liquid or you are giving emulsified antigen, and which route you are taking, intraperitoneal, intramuscular, intradermal, intravenous. So that is also very important. Then charge and valencies, they are related with the chemical structure. Then enzyme susceptibility, it is not very important. The very important thing is optimum concentration. The antigen given in optimum concentration generally is very important, suitable immunological reaction or optimum immunological reaction. This phenomenon is known as signal discrimination. That means antigen, if it is given in low quantity, so there is no response because antibody can and other immune factors can suppress the antigen. Okay? So there is non-responsiveness, and that non-responsiveness is known as energy. Energy, A-N-E-R-G. Okay? If it is given in optimum concentration, it definitely leads to immune response. If it is maximum concentration, that maximum concentration leads to again to non-responsiveness and energy, and sometimes it is in the form of hypersensitivity. Okay, so this phenomenon is known as signal discrimination. That means optimum is immunity, low and high, they leads to unresponsiveness or allergic state in the individual or in the organism. Okay, got it? Now the last one. What happens? Signal discrimination. Okay. Now the last one, that is factors. What happens? You want to ask something? The last one, that is factors that influence the immunogenicity of proteins. These are parameters. If size is large, so their antigen increase immunogenicity. So large size of antigen can increase its antigenicity. If it is small, then can increase